Good day, this is Professor Will, CBMD PhD. Today is Monday, June 21st, 2021. It is 2:37 and 58 seconds, a.m. Eastern Standard Time. Today's research, new imaging techniques. And don't forget to check out my website. So let's hop to it here. Imaging at the tip of a needle. Date June 18, 2021. Source, University of Exeter. Researchers developed a new technique that can revolutionize medical imaging procedures using light. Researchers pioneered a new way in which to control light that has been scrambled by passage through a single hair-thin strand of optical fiber. These ultra-thin fibers hold much promise for the next generation of medical endoscopes, enabling high-resolution imaging deep inside the body at the tip of a needle. Conventional endoscopes are millimeters wide and have limited resolution, so cannot be used to inspect individual cells. These single optical fibers are approximately 10x narrower and can enable higher resolution imaging, enough to examine the features of individual cells directly inside living tissue. It is normally only possible to view cells once they have been taken outside the body and placed in a microscope. The catch is that we can't directly look through optical fibers as they scramble the light sent through them. This problem can be solved by first calibrating an optical fiber to understand how it blurs images, and then using this calibration information as a key to decipher images from the scrambled light. Researchers developed a way to measure this key extremely rapidly, compressively sampling the optical transmission matrix of a multimode fiber. However, the measured key is very fragile and easily changes if the fiber bends or twists, rendering deployment of this technology in real clinical settings currently very challenging. As researchers overcome this problem, the Exeter-based team have now developed a new way to keep track of how the image unscrambling key changes while the fiber is in use. This provides a way to maintain high-resolution imaging even as a single fiber-based microendoscope flexes. The researchers achieved this by borrowing a concept used in astronomy to see through atmospheric turbulence and applying it to look through optical fibers. The method relies on a guide star, which in their case is a small brightly fluorescing particle on the end of the fiber. Light from the guide star encodes how the key changes when the fiber bends, thus ensuring imaging is not disrupted. This is a key advance for the development of flexible ultra-thin endoscopes. These new imaging devices can be used to guide biopsy needles to the right place and help identify disease cells within the body. Researchers hope that their work brings visualization of subcellular processes deep inside the body a step closer to reality. It helps to translate this technology from the lab to the clinic. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.